Howdy! Today we will be discussing humoral immune responses and the effector functions of humoral immunity. For context, humoral immunity is part of the adaptive immunity that fights off infections through the production of antibodies that are produced by B lymphocytes. Phases and types of humoral immune responses. The process begins when mature naive lymphocytes, B lymphocytes, recognize an antigen through the BCR. The B lymphocyte then activates upon recognition of the antigen and proceeds to proliferate and become an effector cell. Following this process, they also differentiate into plasma cells and memory cells. Stimulation of B lymphocytes by antigen. There are two types of ways B lymphocytes can be stimulated, those being by T-dependent antigens and T-independent antigens. T-dependent antigens are protein antigens that require the assistance of helper T lymphocytes. This response produces high affinity antibodies, memory B cells, and class switching. T-independent antigens are non-protein antigens like lipids and polysaccharides that do not require T cell intervention and leads to the production of antibodies with shorter lifespans, affinities, and memory. Role of innate immune signals in B cell activation. There are two major innate immune signals that enhance B cell activation. This can be through a microbial antigen coded in a C3d. For example, when the BCR recognizes the microbial antigen, the co-receptor complex containing CR2 recognizes the antigen attached with a C3d. Excuse me. When both bind at the same time, the activation signal of the inside of the B cells are then amplified, which lowers the amount of antigen needed to activate the B cells, resulting in a stronger antibody response. Another way can be through the B cell receiving a signal through its TLR or toll-like receptor. When the B cell receptor binds to the antigen, microbial molecules known as PAMPs bind to the TLRs, also inducing an additional activation signal, which can promote B cell proliferation and antibody production.
Heavy chain isotope class switching. This is when the B cells alter their constant region from an IgM antibody in order to form other antibodies like IgG, IgA, and IgE. It requires CD40 on the B cell and cytokines from T helper cells. This gives specific antibodies to specific pathogens. Affinity maturation is a way, sorry, antibodies are able to increase their binding site strength to antigens. The first step is for the B cell to rapidly mutate. The B cells with the best binding genes will go into the germinal center to become either plasma cells or memory cells. As you can see here, this is a weaker uh, antibody, and then it becomes stronger binding sites. Regulation of humoral immunity antibody feedback. This is a mechanism that stops B cell activation, reducing antibodies. The antibody will first bind to an antigen, and the antigen antibody complex will then bind to the BCR and to the FC receptor, which is right here, which will cause the B cell to stop the activation of secreting antibodies. This prevents autoimmunity diseases from occurring. Effector functions of humoral immunity. There are 5 effector functions. The first one is neutralization. This is where antigens on the epithelial surfaces, such as the gastrointestinal tract and the skin, will bind to microbes which prevent infection. Number 2 over here, we have opsonization and phagocytosis. This is when the antibodies will bind to the microbes as seen in this photo, tagging it for phagocytes to come on over and engulf the microbes. Complement activation. This is when antibody binded microbes will start up the classical complement pathway. And for finally the last one, oh no, sorry, not the last one, fourth one. This is ADCC. This is when antibodies will bind to infected cells, usually viral, and natural killer cells. FC receptor will bind to the antibody, causing the NK killer cells, the natural killer cells, to then attack and kill the infected viral cell. Finally, for the last one, for mast cell and eosinophil activation, IgE will bind to the mast cell slash eosinophil. The antigens will then get cross-linked in IgE, which will release mediators. This is essential for parasite and allergy defense.
Mucosal immunity protects the gastrointestinal and respiratory tracts. In these tissues, plasma cells are going to produce IgA antibodies in the lamina propria, and IgA is transported across epithelial cells by the poly-IgA receptor. Once released into the lumen, IgA binds ingested or inhaled microbes and prevents them from attaching to and crossing the epithelium. This prevents infection before microbes can invade underlying tissues and serves as an important first line of defense at mucosal surfaces. Neonatal immunity protects newborns through maternal antibodies. During pregnancy, maternal IgG antibodies are going to cross the placenta by binding to the neonatal Fc receptor, or FcRn, and enter the fetal circulation. After birth, infants are going to receive additional antibodies through colostrum and breast milk. In particular, IgA is going to provide mucosal protection in the infant's gastrointestinal tract. As a result, newborns are going to acquire passive immunity and gain protection against microbes to which their mothers were previously exposed to or vaccinated against. Many microbes have evolved ways to evade humoral immunity. One mechanism is antigenic variation. This is where microbes are going to change their surface antigens, so existing antibodies can no longer recognize them effectively. Examples include influenza and HIV. Another strategy is the inhibition of the complement activation, which is going to reduce complement-mediated destruction of microbes. This can be seen in Staphylococcus. And some bacteria, like Streptococcus, also produce a hyaluronic capsule, and this is going to help shield them from antibody-mediated immune responses. And all three of these mechanisms are going to increase the microbial survival and make immune elimination more difficult. Vaccination is the process of stimulating protective adaptive immune responses against microbes by exposure to non-pathogenic forms or components of microbes. Attenuated vaccines are going to contain microbes that have been treated to abolish their pathogenicity while still retaining infectivity and antigenicity, seen in the MMR. Subunit vaccines are composed of microbial proteins and polysaccharides. This is for the HPV. Conjugate vaccines are going to chemically couple microbial polysaccharides to proteins so that helper T cells are activated and high affinity antibodies are produced, seen in the pneumonia. Synthetic or recombinant vaccines are going to use protein synthesized in the laboratory, and this is seen in the hepatitis B. Viral vector vaccines are going to incorporate microbial antigens into viral vectors, which infect host cells. Examples include the Ebola and COVID. DNA vaccines use plasmid DNA encoding a microbial antigen, and this plasmid is taken up by APCs, which then produce the antigen. And the mRNA vaccines deliver messenger RNA that's translated into the antigen by host cells, and these last three are all seen in the COVID vaccines. I worked on slides pertaining to the protection at barrier surfaces and in newborns, microbial survival strategies, and vaccine-induced immunity, and I just used a textbook.